Are there any long-term ethical issues with this? So, for example, let's say we have the ability not just to fix a genome so you're less susceptible to a disease, but so that you can get new traits altogether. Can we breed people to specifications, and would that be a good thing?
Well, that's obviously a controversial area of genetics. I think no one will argue with curing horrible diseases through gene therapy or other, or catching disease early and figuring out how to deal with it. But modifying behavioral traits, modifying things that you're talking about enhancements of someone's physique or what have you. I think that's a very controversial area, and one that one would go through that process very slowly, shall we say, to be able to determine what's proper ethically.
And importantly, that's a societal decision. I think some of these technologies may make these things technically feasible, but it's just because it's technically feasible doesn't mean you should do it. And also, just because it's technically feasible doesn't mean that the technologists should be the ones deciding if it's right or wrong. So for those kinds of questions, it's all voices that need to be heard, and there has to be a societal discussion about where we draw the line, if we draw a line, at manipulating genomes.
Okay. Because there's a possibility that everybody might be typed at birth, and somebody might look at your genome at birth and say, well, doesn't look like you're going to amount to much.
I'll go one step further. People will probably get their genome sequenced before birth so that you can predict, because now there's DNA circulating in pregnant women that has the fetal DNA, and in principle it's possible to sequence their DNA before birth. So you'll actually be able to predict the sorts of diseases they might be at risk for or might have at birth, and you'd be able to catch them early.
And if I could even push that further, Mike and I are aware of companies and technologies where you can take an embryo, you can take one cell from the embryo. It turns out that if you do this early enough in development, it doesn't kill the embryo. Just that one cell it can be made up for, and then you can sequence and make measurements on that single cell to learn things about this embryo that is now an 8-cell embryo, just 2 or 3 cell divisions since the moment of conception. It sounds amazing, and it is amazing, but there are companies that are doing this.
Is there any possible downside to this? Is there any way in which this knowledge might turn out to be harmful if it's misused?
Absolutely. I mean, I think a corollary to the technologies that we develop can make amazing saves, if you will, is that if used improperly, you could imagine targeting soldiers from the other side who might have certain genetic features and saying, we're going to build viruses that are specifically designed to attack those people, all kinds of things that you can imagine. And again, I think society needs to decide if and where to draw the line. The problem is sometimes the bad guys, if there are bad guys out there, might want to have a different set of motives and operating principles. So I do think that this is a powerful technology. We're obviously committed to using it for the benefit of humankind. That's our #1 priority. But I think it would be putting our head in the sand to say it's impossible for somebody to use it for nefarious goals. I don't know if you agree.
No, it's true. I guess right now I think we're certainly very focused, as Russ says, on using it to improve human health for the benefit of people. To me, it's a no-brainer. This is what we should be doing. But like all things, you should act responsibly and accordingly. The area will be regulated, no doubt, to try and minimize these sorts of things, but you do have to watch for abuses.
Does it look like there's a pretty open field, like this research is progressing nicely, no major obstacles in the way, got enough funding, and you just see a steady progress until all of the goals are achieved in the not-too-distant future?
Well, there's still many challenges ahead. We are not perfect at interpreting the genome. It's still a huge challenge to get to the goal I mentioned, where if a base change occurs, if there's a change in your DNA at one spot, what will it be its consequences? That's going to take quite some time to be able to sort that out. It's also going to take a lot of resources, even much more than we have now. So there still are funding challenges for many of us. We'd like to be able to enhance what we're trying to do. It's an incredibly exciting area. It's incredibly fast-moving, but it's going to take a lot of effort to be able to get to the point I mentioned. I also think we want to get at this issue Russ raised earlier about not only understanding your DNA, but how it interacts, if you will, with the environment. So that, for example, should you be a pesticide worker if you're at risk for Parkinson's? The answer is no, based on some interesting studies from one of our colleagues at Toll Butte. But we want to understand that better. Which kinds of things should you do and should you not do based on your DNA? And that's going to take a lot more research. It's a very open frontier, a huge challenge ahead.
You know, I'm interested in statistics and computer science, and another big challenge is there are variations in the genome that are only going to be present in 1, 2, 10, 50 people, very rare. It's going to be impossible to study those statistically. We like to take 1,000 people or 10,000 people with a variation and compare them to 10,000 people who don't have the variation and learn the differences. That has worked well, but when you're talking about a rare familial or even personal variation, those kinds of statistical analyses are not going to be possible, and then we're going to have a much deeper understanding of how to predict these through computation. And so there's a huge opportunity and challenge for computational analysis of rare DNA variations for which we're never going to have enough examples to do it in a straightforward statistical way.
So I think your research is mostly computer modeling, right? You're modeling the behavior rather than doing wet work?
My research, absolutely. We do not have, the wettest thing in my laboratory is the espresso machine.
Okay.
And everything else is in the computer. We have cluster computers. We have, all kinds of computational software. It's been an amazing time because when you have 3 billion bases or then 1,000 people with 3 billion bases, this is fundamentally an information processing problem. It's why it's exciting also to be here in Silicon Valley where those kinds of numbers don't scare people.
Now, if your genome makes you susceptible to a disease, does that mean that you have a faulty gene, there's something wrong with it, or there's a normal variation, and so different people are normally responsive to different things, so one individual might suffer, but somebody will be resistant, so the species as a whole will be hardy and able to survive?
Well, that's what it's thought, that a lot of these variants, if you will, are common, and so some of them help certain groups survive adverse conditions and others are deleterious. And the classic example is sickle cell anemia, where individuals with a certain genetic trait that makes them susceptible to sickle cell anemia, they're actually more resistant to malaria. And there are many other examples of that as well. So some of this stuff can be protective, but and then some of it's then deleterious in the wrong situations.
One of my favorite examples on that note is diabetes. So we know that there's this problem with obesity, and obesity is leading to an increased epidemic of type 2 diabetes. Why is it that so many people get obese and so many people get diabetes? Well, 5,000 years ago, when droughts happened, if you had an ability to retain calories and get the maximum efficiency of the food you ate, then you were the one who survived at a time of drought or famine. That was an incredibly valuable capability. Now, fast forward to the era of McDonald's and plentiful food, that's not the greatest adaptation to have in terms of your ability to cling to calories because there's too many calories around. And so diabetes probably, and there's been work in this area, arose as a very useful way to avoid starvation, and now we're left with the consequences.
So whether your genomics work for you might depend on the environment. Maybe you're well defended against something that's never going to happen in your environment.
Exactly.
Yep.
Exactly. And so you always have to think back. As a geneticist, we do this a lot. What was it like 10,000 years ago as a hunter-gatherer, say in Africa or in America, and what were the big things? Starvation and infection. So much of our genome has been optimized to not starve to death and to avoid dying of an infection. Again, fast forward, we have antibiotics, we have a lot of food. It's a different set of issues now that are killing us. And we have not evolved to respond to these new threats.
We have about 30 seconds left. Any last comments?
Well, I want to say the other area that genomics is having a huge impact that we didn't talk about is cancer and undiagnosed disease. And so I think these are areas that if you have cancer, every cancer is different. You will, in fact, want to get your genome sequenced to be able to get the right drugs for the cancer. And genomics is also having a huge impact in undiagnosed disease as well for trying to solve some of these. But there's huge challenges ahead.
Those are here today. I agree entirely. That's what we can do today.
Okay, great. And we're going to have to leave it at that. I'd like to thank my guests, Michael Snyder and Russ Altman. Thank you for watching. I'm Marty Wasserman. Visit our website, www.futuretalk.net. See you next time.